DNA and Protein Synthesis: How Our Traits Are Determined, by Ashley Murphy. DNA, or deoxyribonucleic acid, contains the instructions for everything that occurs within our bodies, even our traits. It makes up our chromosomes and is stored in the nucleus in every cell of our body. DNA structure is called a double helix because it is made up of two strands of nucleotides. The bases found in DNA are adenine, thymine, cytosine, and guanine. The bases in DNA pair up in a particular way. Adenine will pair up with thymine, while cytosine will pair up with guanine. Whenever our cells divide and multiply, DNA needs to make more copies of itself. This process is called DNA replication and occurs in the nucleus. DNA contains the information to determine our traits, like hair color and eye color. In order to determine your traits, DNA must first be transcribed into mRNA and translated into a protein. The shape and structure of the protein will determine your traits. This process is known as protein synthesis. Before we discuss the process of protein synthesis, we need to review RNA. RNA, or ribonucleic acid, is what DNA is converted into in order for the message DNA contains to leave the nucleus and travel to the ribosome. RNA is a single strand of nucleotides composed of adenine, uracil, cytosine, and guanine. DNA and RNA have a few differences. DNA is much larger than RNA. DNA is a double helix while RNA is a single strand. DNA contains the bases A, T, G, and C, while RNA contains the bases A, U, G, and C. Finally, the sugar in DNA is called deoxyribose, while the sugar in RNA is called ribose. Now we will focus on the process of protein synthesis. The first step is transcription, where DNA is transcribed into mRNA. Transcription occurs in the nucleus of the cell. The double strand of DNA is separated by RNA polymerase, and then the bases of DNA are paired with the corresponding RNA bases. An A in DNA will bind to a U in RNA. A T in DNA will bind with an A in RNA. A G in DNA will bind to a C in RNA, and a C in DNA will bind to a G in RNA. After a strand of mRNA is made, it will travel to the ribosome to make proteins. The process of RNA changing into an amino acid sequence, or a protein, is known as translation. Translation occurs in the ribosome of the cell, which is responsible for making proteins. The tRNA molecule will attach their anticodon to the matching codon on the RNA strand. As the ribosome moves down the strand, the tRNA leaves behind an amino acid, which links with other amino acids. This chain of amino acids forms the protein. A closer look at the tRNA shows the amino acid at the top and the anticodon at the bottom. A codon, which can be found with the RNA strand, is a set of 3 nitrogen bases. The first codon you should look for is AUG because this is the start codon. You can remember this because school always starts in August, which is abbreviated AUG. After you find AUG, you need to group every 3 bases to make a codon. Once you make your codons, you will use a codon chart to turn them into a chain of amino acids. There are a variety of charts we'll use, but the most common are the ones found here. Translation will continue until a stop codon is reached. This signifies translation has stopped and your protein is complete. When replicating a strand of DNA, it's important to remember the base pair rule. A will bind to T, T will bind to A, G will bind to C, and C will bind to G. When transcribing DNA into RNA, it's important to remember the base pair rule. An A in DNA will bind to a U in RNA, a T in DNA will bind to an A in RNA, a G in DNA will bind to a C in RNA, and a C in DNA will bind to a G in RNA. When translating DNA into RNA, you should find AUG first and then group together every 3 nitrogen bases. Don't forget to use your codon chart to translate your codons into a chain of amino acids, also known as a protein. Sometimes changes in DNA do occur. These changes are called mutations. Since DNA contains the information that determines your traits, a change in DNA can affect your traits. Some examples of mutations include substitution, where a base is switched with another one, a deletion, where a base is removed, or an insertion, a base is added. Not every mutation will lead to a change in your protein because several codons code for the same amino acid. But if there is a change in the amino acid, a change in your protein and traits will occur.